So now let's start with this chapter, gastroretentive drug delivery system. So before we move ahead with the what are actual gastroretentive or how to actually make GRDDS formulation, let me give you a brief background and an introduction about that. Okay. So there are many drugs that are currently available in the market which shows poor absorption in your lower gastrointestinal tract. So lower region of your gastrointestinal tract, these drugs they tend to show poor absorption. And that is why we need something that keeps them floated or keeps them retained in a particular area so that they can be absorbed in a proper manner. Okay, so basically the aim of this GRDDS formulation is to make the formulation available in the stomach area or to make it available either in the upper region of small intestine. Okay. In addition to this, so that was one of the idea behind formulating GRDDS. That was the drugs which has poor absorption, we can help them to get absorbed better. Second rationale could be behind making GRDDS is there are some drugs whose potential site of action itself is stomach. Okay. For example, there are drugs which we give against H. pylori infections. Okay. So such drugs requires that they should be absorbed only from the stomach region. So for those drugs, GRDDS is a very good drug delivery system. Now, over a period of years, we have made several attempts so that we can make a good GRDDS system or gastric retained drug delivery system to overcome the limited regions and time for absorption required in the GR tract. Okay. So what we basically do is we somehow try to retain that drug in the GRDDS by either of the two options. Okay. What we do is either we make the drug or the formulation such that it is floating on the stomach fluids or gastric fluids that are present in the stomach area. So either the system is floating on that surface, because of which it is going to be retained in that particular area, or we make that system in such a manner that it is an effervescent kind of system. So initially its density is high when it is in the solid form, but the moment it comes in contact with water that is present in your stomach area, it starts releasing effervescence, and because of that effervescence, it tries to float on the fluid that is present. Okay, so either you can work with density basis, that is floating and everything, or you can make it with respect to size basis. Okay, now what is the rationale behind making it on the basis of size? Now our stomach is attached to the small intestine with the help of a particular sphincter that we will see in the coming slides. Okay, so that region from where even your food or your drug or your tablet, whatever it is, when it enters from the stomach region to the small intestinal region, it has to pass through that narrow region. Okay, so this narrow region, the scientists have taken advantage. They have made formulations which are bigger than this diameter of this narrow region. Okay, so in this entire process, what happens is because the diameter is bigger, it takes time for that particular dosage form to pass from the stomach region into the intestinal region. And in this entire process, your formulation is getting retained in the specific region. Okay, so this is another approach by which you can make a GRDDS tablet or GRDDS dosage form. Okay, There is another approach that is being recently tried. So what they do is they make microparticles, and they make sure that these microparticles are mucoadhesive in nature. So all our gastrointestinal tract is coated by a thin layer of mucoadhesive substance, mucin. Okay, so these microparticles, when you take it through oral route, they go and attach to your gastrointestinal tract. They go and actually stick like a glue to that mucoadhesive patch. Okay, so in this attempt also, your particles are retained in the gastric area. They do not go into the intestinal area. However, this system has a little drawback because they are very small particles. Even a slight movement of the stomach because of the peristalsis, etc., there are chances that these microparticles might get detached from the surface and they might enter into the small intestine. Okay. Another issue with this system is, out of the total bulk of the formulation or the dosage form that you give, around almost 2 to 3% only gets attached. Rest all enters into the small intestinal region. Okay. So even though a good system, you have to assess its pros and cons, and then you have to administer it to the patient. Okay. So this was the brief background of gastrointestinal drug delivery system. The major player in gastrointestinal drug delivery system is the food intake. What kind of food that person has before he takes a GRDDS dosage form, that will determine how that GRDDS is going to act. Okay. Do you have any idea about what is gastric retention time?
Ma'am, the amount of time a dosage form remains inside the stomach.
Okay. So any idea what is gastric retention time of stomach?
I guess it is 2 to 3 hours.
It is around 2 to 3 hours. Yes. So gastric retention time is basically the time the food takes, or any material which you ingest, it takes to enter from one compartment to another. So for if you take this gastrointestinal tract overall, the food is going to remain for a particular time in the stomach area. It has to the certain juices of your stomach are going to act on it. The food is going to get broken down into small particles. So basically the function that is carried out in the stomach is breaking bigger particles into smaller particles of a smaller particle size. After that particular work is done, the food will now enter into the small intestinal region. After that, it is going to enter into the large intestine. Okay. So when the food passes from one region to another, it is called as its retention time, or it's called as its transit time. Okay. So the goal of GRDDS system would be to maximize the transit time of the dosage form, so that for the maximum time it retains in the stomach area, and once its function is over, it then enters into the small intestine.